Hello, everybody. Welcome back. So today's video is going to be all about enzymes. So that is going to be the title of our notes today. And enzymes, I have mentioned before when we talked about proteins, and that's because enzymes are a protein. And they are probably the most important protein in our cells. So let's write that down. So what I'm going to do is I'm going to go through a list and I'm just going to describe what enzymes are and what they do. And that's all this video is going to be about is like, what is an enzyme and what does it do? So first of all, you know this already, enzymes are protein. So they're made of, I'm not going to say they're made of protein, they are a protein. Enzymes are a protein. And I'm just going to say real quickly that you have DNA in your cells. Your DNA is a code for all the proteins. Enzymes are probably the most important protein. So basically, your DNA is a code for all your enzymes. Okay. And I just want you to think back about what a protein is. It is a string of amino acids that folds up into this very specific shape and it ends and makes this little molecular machine. And that's what an enzyme is. It is a molecular machine. And so I've mentioned this before that enzymes are the machines that perform chemical reactions. So enzymes are molecular machines that perform chemical reactions. Oops. And so in your cells, there are chemical reactions happening all the time. And they don't happen randomly, they happen because of the enzymes in your cells. So if there is a chemical reaction that is needed, then there will be an enzyme produced, and that enzyme will do that chemical reaction. Okay, and I've mentioned this before that there are two kinds of chemical reactions. Okay, so let me just do a little bit of a review. And we talked about this already in a previous video. There are two kinds of chemical reactions. There is one called a dehydration, and that is when you have two pieces that come together to make a bigger piece. So dehydrations are reactions that are building bigger molecules. So I'm just going to put building bigger molecules. And the opposite is called hydrolysis. And hydrolysis reactions are when bigger molecules are being broken down. So breaking down molecules. Okay, and so in our bodies and all living organisms is all about making things and then breaking them down into pieces, using those pieces to make things, breaking those down into pieces, using those pieces to make things. So it's like this recycling of chemicals constantly. That is what life is all about. And so in your cells, you are constantly making bigger molecules from pieces of molecules that you broke down. So there's this recycling happening constantly. Okay, so this is the way that the enzymes work. What they are this very large protein. Okay, and so I'm going to just draw kind of a funny picture of a very large protein. And I'm making it kind of bubbly because if you remember, they're made of amino acids, and we sometimes represent amino acids in this like balls. And so let's say this is a very large protein and it's an enzyme. So I'm just going to put large protein, and this is our enzyme. Whoops. Hang on, let me just respell that. IN. and this is an enzyme. Okay, so all enzymes have properties. Okay, one very important property that all enzymes have is they have this hole. There is a little pocket that fits another chemical. Okay, so a protein is a chemical and is large, made of amino acids, and it's got this little hole. This little hole has a special name called the active site. And the active site is very specific in shape and holds a very specific molecule. This is the molecule that it's going to do the chemical reaction on. So either it's going to be a very large molecule that is going to break apart into two pieces, or it's going to have two little pockets to hold two molecules to bring them together to make a bigger molecule. Okay, so it just depends on the enzyme. So the active site, I'm going to say, has a specific shape to hold a molecule. And if a molecule goes into an enzyme, it has a special name. It's called a substrate. So I'm just going to put this in parentheses. It's called substrate. Substrate is just a name saying that it is a molecule that fits into an enzyme. So we have this large protein, has this little pocket that we call the active site that holds the substrate. This active site has a very specific shape that matches the substrate. And every enzyme can only hold one substrate. Okay, you can't have one enzyme do a lot of different chemical reactions. Every enzyme can only do one type of chemical reaction. And that is very important. So I'm going to write that down. So I'm going to say every enzyme can only do one type of chemical reaction. Okay, and as you can imagine, there are millions of chemical reactions happening in your cells all the time. So you are going to need millions of different enzymes to do all the different chemical reactions because every enzyme can only do one kind of reaction. Okay, another point that I just want to add here is that the enzyme itself doesn't change. Okay, the substrate that goes into the enzyme absolutely changes. It either gets broken apart or it comes together with another piece, and that will change. But the enzyme itself doesn't change. And in that way, that enzyme can then do another chemical reaction and then another and then another. And that's typically what happens is one enzyme will just do chemical reactions over and over and over. And in that way, the enzyme themselves are being recycled. So I'm going to write that. Enzymes don't change, like they don't change shape. And therefore, and can do many chemical reactions over and over. Okay, so for example, it will do a chemical reaction and then it will find a new substrate, do the chemical reaction again, find a new substrate, do the chemical reaction again. Okay, let's see. So let me show you a little cartoon picture just to maybe clear some of this up in case you're a little bit confused. Let's see here. So let me show you a So I'm going to show you a cartoon picture of an enzyme. And this blue part right here is an enzyme, okay? And it's just a cartoon picture. And it's a protein made of amino acids, and it has a certain pocket right here that can hold a certain substrate, and that pocket is called an active site, okay? And then it will do its work, the little machine, and then it will take that piece and make it into two pieces. So that is a chemical reaction. It's called a hydrolysis, which means you had a big molecule and you broke it into two. So you're breaking down molecule. So this is an example of what an enzyme can do. So here, the blue thing is an enzyme, the red thing is a substrate, Then they come together in an enzyme substrate complex. And then when the enzyme's done doing its chemical reaction, it spits out the products. And now we've got something new. Okay, but the enzyme itself is not different. And I just want to show you one more picture of the opposite reaction of a dehydration reaction. Okay, so that first example was a hydrolysis. And this is a dehydration where you have an enzyme that's going to take two pieces and make it into one piece. Okay, so let me just write a little label here. So this is a hydrolysis, and this is a dehydration. Okay, and those are the only two possible chemical reactions possible, and enzymes can do one or the other depending on what the enzyme is. Okay, so in this picture, we've got the blue enzyme, and it's got two separate pockets where two pieces come into, and then the enzyme does its little work, it does its job, and makes it into one product. Okay, and so then it releases. So dehydration is when you have multiple pieces made into one. Hydrolysis is when you have one piece, make them into two. Okay. Okay, so that's what an enzyme does. And there are lots of different enzymes. And I just want to show you another picture that shows you a real example of a lot of chemical reactions that are happening. Okay, so this is called a metabolic pathway. Let's see, where is that picture? Oh, right here. Metabolic pathway. And this is just showing you a real example of why we're learning this. This is why we have to know what this is. So this is kind of a complicated picture, but I just want to point out that every arrow represents the chemical reaction that is taking place. On the arrow, normally we write the enzyme, okay? So I just underlined the enzyme here. This one is called aconitase. Okay, so aconitase is the name of the enzyme. It's this large protein. It's going to take this molecule, which is called citrate, and it's going to make it, it's going to do a chemical reaction on it. And now citrate has been changed into cis aconitate. That's a new molecule. And this aconitase is going to take cis aconitate and do something to it to make it isocitrate. And then this enzyme here is called isocitrate dehydrogenase, and it's going to take this molecule, do something to it to change it to ketoglutarate. Okay, so my point is the substrate goes into the enzyme. So this is, let me circle some stuff here. So I'm going to do hot pink. This is the substrate, goes into this enzyme, and that enzyme changes that substrate into a product. That product is now the substrate to the next enzyme. The enzyme is going to change it, and this is the product, which is now the substrate to the next enzyme. So this is the way metabolic processes happen in your cells. You have these enzymes that are going to take a molecule and change it. The enzyme itself doesn't change, and it's going to continue to do that same chemical reaction over and over. Okay, so now that we know what enzymes do, I just want to mention that enzymes are affected by some factors. There are some factors in the environment that can either make an enzyme better or make an enzyme worse. Okay, and when these factors are sometimes used in your body to either speed up chemical reactions or to shut them down completely. Okay, so I'm just going to make a new list here. This is called factors that affect enzymes. Okay, and maybe this is an A. I always get confused about affect or effect. I'm not totally sure. Maybe one of you can correct me on that one. But these are some factors. I'm gonna make a list. I'm gonna number the list, and these are things that can either speed up an enzyme or slow down an enzyme or make an enzyme better. And so the first thing I want to point out is that every enzyme has an optimal temperature. There's a temperature that an enzyme loves and works best at. Okay, so I'm going to just put temperature as number one. Okay, and so as humans, we have a body temperature, and our body temperature is 98.6 degrees Fahrenheit, which is 37 degrees Celsius. And the reason we have this temperature is because our enzymes work best at that temperature. So our body has to keep our bodies at that temperature or close to it for our enzymes to work. If we get too cold, the enzymes slow down and they can't work efficiently. If our enzymes get too hot, the enzymes fall apart and they get denatured, and now you die. So that is essentially why we have to keep our temperature within a very strict range. If you get too cold, your body will warm yourself up. If you get too hot, your body will cool yourself down to stay in that temperature so that your enzymes are working effectively. Okay? So this is called an optimal temperature. And for humans, it is 98.6 degrees. Okay? So temperature, enzymes, I'm going to say human enzymes work best optimally at 98.6 degrees Fahrenheit. Okay. Different organisms, depending on where they live and how they evolved, have different optimal temperatures. So there are some animals and some other organisms like bacteria that have to be in the cold. Okay. So those enzymes work best in the cold. Some organisms and some bacteria work best in the very hot and have to be in the heat. So there are some bacteria that live in these thermal vents that have to have it hot or they don't work. Okay, so just depending on what organism you are. Okay. Similarly to temperature, there is this concept of pH. Okay, so pH means how acidic or basic your environment is. And our enzymes have a very specific pH that they have to be at. And again, your body will maintain that pH for your enzymes. Okay, so human enzymes work best at a specific pH. For us, it is pH 7, which is neutral. Okay, so I'm going to say most human enzymes work best. Oops. Um, at pH 7. Okay, there are some enzymes in your stomach that work best at a lower pH, like pH 2. Okay, so I'm going to say stomach enzymes. And you have enzymes in your stomach to break down food that you eat. And these work, and your stomach pH is at pH 2, which is very acidic. Okay, and so those enzymes have to be at pH 2 or they don't work. And I just want to point out that what pH measures is how many hydrogens are around. Okay, the lower the number, the more acidic. So pH 2 is acidic, pH 7 is neutral. Okay, and so those are two factors that affect your enzymes. Okay, what else? Okay, the last one is inhibitors. Okay, this is the last one. So all enzymes need to be able to turn themselves on and off. You have enzymes in your cells, and sometimes you don't necessarily want them doing chemical reactions like constantly, because then you might make too many products and then you'll have this buildup of products and it's too much. So, for example, you're not going to want to go, like, let's say when you go to the grocery store and you buy a gallon of milk, you're not going to want to go buy a gallon of milk every single day because it's too much milk, right? And then you're going to have too much milk. You're not going to have room in your fridge. It's just like a waste, and you're going to throw it away. It's a waste of energy and resources. So you need to be able to shut down enzymes sometimes. And so the way that your body does that is through inhibitors. There are two kinds of inhibitors. There is one that is called a competitive inhibitor. Inhibitor. Oops. Okay. And a competitive inhibitor is one where there is a specific substrate that fits into the enzyme, but there is another molecule that has that same shape of the substrate that essentially goes into the active site and blocks the correct substrate. So let me draw a picture and then I'm going to write down what the explanation is. So let's say this is our enzyme and this is our active site. Let's say the normal substrate looks like this. It fits perfectly into this active site. Sorry, my picture is not very good. Let me redraw it. So it's got to be right, perfect. It's got to be the right shape. Okay, let's say that's the correct substrate. Okay, so this is the enzyme, and this purple thing is the substrate. So let's say we have another enzyme, and there's this other molecule that actually has the same shape as the active site, but it's a different molecule. So but it fits in the active site. So it'll go into the active site and it will just stay there. And the enzyme can't do a chemical reaction on it because it's not the correct substrate. But what it does is it kind of plugs up the active site so that the normal substrate can't fit and in a way blocks the substrate. So I'm going to put inhibitor here. Okay. That's supposed to be an H. Okay. So a competitive inhibitor blocks substrate. And in a way, this is just how to turn off the enzyme, which sometimes you need to do because you have too much product. Okay, a second inhibitor is called an allosteric inhibitor. And an allosteric inhibitor is the same in that it turns off an enzyme, but it does it in a different way. So I'm going to draw an enzyme here, and I'm going to put another like place on the enzyme. This is called the allosteric site. Okay. Up here, this is called the active site. Okay, so let me put some arrows here. So this is the allosteric site. That's the active site. And the allosteric site is a place in another part of the enzyme. And when an allosteric inhibitor binds to the allosteric site, it essentially closes the active site. Okay, so I'm going to draw another picture of the same enzyme. And what happens is when the allosteric inhibitor is bound to the allosteric site, it causes the active site to close. Okay, so it's like closes the door. So this is a functional enzyme, which means that it can do the chemical reaction that it needs to. This is a non-functional enzyme. It's turned off. Okay, so both of these methods are used naturally to turn off enzymes when an enzyme needs to be shut down. Okay, so that's all for the lecture. Before I go, I just want to show you a picture of a real enzyme, so you can see it. This is a protein, and this is called a ribbon picture. Okay, so this is an enzyme called glucosidase. And over here on the left is the ribbon representation of the enzyme. Okay, it's hard to see, but proteins are kind of a complex thing, and so this is what a protein would look like. This is glucosidase, and what it does is maltose, which is a sugar found in milk, goes into this enzyme, and so when your body is digesting milk, it will take maltose from the milk and it will break it apart into the glucose products. Okay, so maltose is a disaccharide. It goes into the enzyme active site, which is right here in the middle. So it's kind of hard to see, but it's right here in the middle. And then it will take apart the maltose and make it into the monosaccharides, which is glucose. And now your body can use glucose to either make a new molecule or it can actually use glucose to make some ATP. Okay, That's it for today's video. I hope you're having a great day, and I will see you next time. Bye bye.